Global sequence alignment seeks the similarity between the two strings throughoutly. It means that in the global sequence alignment, we have to see the alignment between the two strings from one end to the other end globally. So this is possible only when the two protein sequences that are very similar to each other, and that is only possible if they are very similar to each other. So these proteins, they might be different from each other, and in this case the global sequence alignment score will be very less. So if we want to attain the high score for the two sequences for the proteins, the condition is that these two proteins should be very similar to each other. So these type of proteins these are present within the wide range of organisms that are similar to each other, and it ranges from humans to the fruit flies, and we can find the similar proteins in very diverse organisms. Sometimes it happens that the two proteins that are considered for the global sequence alignment, their score is less as compared to the subsequences that are present within these proteins. If we consider two proteins V and W, it is possible that the subsequence that are present within these two proteins that have the much higher score as compared to the score that is present from one end to the other end of the proteins. So the example of this sub sequence is, we can consider the example of homeobox genes, and these homeobox genes, they are involved in the embryonic development, and these genes are present in the wide range of organisms. But these homeo genes, they are different in various organisms, and the difference is that this homeo gene contains a specific domain that is called as homeodomain, that is conserved throughout the protein that are present in various organisms. But the location of this homeodomain is not conserved within these proteins. In some proteins, this homeodomain is present in the initial part or in the start of the protein, and in some proteins in other organisms it is present at the end of the proteins, and so on. Its location is not conserved. So the problem is how to find the conserved area of this homeodomain and ignore the less similar parts of the other part of the proteins. So this problem was solved by the two scientists, Temple Smith and the Michael Waterman, and they have proposed the modified version of the global sequence alignment dynamic programming algorithm, that is the local alignment program. So here is the example of local sequence alignment. Now we can see here we have two gene sequences that is present in two different organisms, and we can consider it as also as a haplotypical genes. One is V and the other is W. In the first gene, we can see in red is the nucleotides that is conserved in this hypothetical gene, and the same domain that is present in the other gene is present at the end of this gene. So we can see the sequence of this domain is CAGT-TT-GT-CAG, and this domain is identical in other gene that is present at the end of the gene. So the conserved sequence that is present in the start of this gene and the end of this gene is this conserved domain. So if we consider the global sequence alignment as we are seeing in this case, we can see that these two domains, they are very far away from each other, and the scoring matrix that we have used, they cannot align these two domains, because in the global sequence alignment we have to consider these proteins starting from nucleotide 1 up till the end of the gene. But if we align these two domains that are conserved in these two genes, then we can see that the scoring matrix for these two aligned genes will be higher as compared to the global sequence of these two genes. So for this purpose, we have to align these and slide these two genes so that these two domains should align with each other, and we can see that these are aligned with each other, and the score will be much higher as compared to the score that is we are considering for the global sequence alignment. So in this case, we will consider only this part of the gene, that is the conserved domain, and because we are only considering the small part of the conserved domain, so this sequence will be called as local sequence alignment. So this is the graphical representation of this local sequence alignment, and we can see here that the conserved domains are identical and cover the only 1/3 of the entire length of the nth gene. So the path from source to the sink will include approximately 2,3 by n, and that is the horizontal edges, and the 1/3 n will be the diagonal edges, matches to corresponding to homeo domain, and the 2,3 by n is the vertical edges. So in this graphic representation, and we can see that this gray line is showing the global sequence alignment, and it starts from source and it ends up to the sink, and we consider the whole sequence of the two genes from starting to the end point, and we can see only the diagonal positions, they are aligned between the two genes. These diagonal positions are matches between the two genes, but the vertical and the horizontal lines, they do not have the matches. They are either mismatches or the indels. But in the case of the local sequence alignment, that is represented by this, we can see it is all single diagonal line. It means that all the nucleotides that are present from this position up to this position, and from this position to this position, they are totally matches with each other. So there is no penalty in this local alignment, and all the nucleotides, they are matches, and they will awarded with +1 score for all of these matches. So we can see very easily that the total score for the global alignment will be much less as compared to the local alignment score.